Hello and welcome to this introduction to microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the emergence of different microorganisms on Earth and how microorganisms have helped to shape us and the world around us. Now, the field of microbiology connects us to some of the very earliest life on Earth, which is thought to have emerged around 3.8 billion years ago, and this is about 750 million years after the Earth had formed. And it's a strange idea to imagine, but for that entire 750 million year period, Earth was completely sterile where no life existed at all. And the reason for this comes down to the conditions on early Earth. You see, primitive Earth was extremely hot, lacked many of the key ingredients needed for life, and was essentially a primordial soup of simple precursor chemicals. However, over time, these chemicals reacted with one another, forming molecules. Now, it might seem a bit far-fetched, but experiments like the famous Miller-Urey experiment have shown us that under conditions thought to mimic early Earth, so high heat, limited atmospheric gases and energy from electrical storms, these simple molecules could combine to form more complex compounds. And from this point, step by step, these compounds gave rise to the first self-replicating systems, initially RNA, which was superseded by DNA, and eventually, after hundreds of millions of years of gradual chemical evolution, the first true cell appeared, and with them, life on Earth had begun. Now, this cell from which all life on Earth is thought to have evolved from is known as the common ancestor, and we can see how much this has changed by looking at the tree of life. We can see a visual representation of how all living things are related to one another based on similarities and differences in their genetic code. And so it all began with a common ancestor, the first cellular life on Earth. From this point, life began to evolve and diversify, and very early on there was a major evolutionary split. This gave rise to two distinct types of cell. The cell at this single point of origin is often referred to as LUCA, the Last Universal Common Ancestor. Now, LUCA wasn't the very first life form on Earth, but rather the most recent ancestor from which all living things today have descended from. And so from this early division, two major branches appeared, the bacteria and the archaea. Both groups consisted of single-celled organisms, many of which were and still are well adapted to the extreme environments of early Earth. Together, these two groups are often described as the prokaryotes. Then much later, so around 2 billion years ago, as the environment became more stable, a third branch emerged. These were cells with a more complex internal structure, giving rise to the eukaryotes. From this group came the great diversity of complex life, everything from fungi and yeasts to plants and eventually animals like us. Now, in microbiology, we also consider several acellular entities such as viruses and prions. These agents lack fundamental characteristics of living organisms, including metabolism and the ability to reproduce independently, and as such, they do not fit within the tree of life. Instead, they exist outside of it, highlighting the distinction between cellular life and these non-living biological agents. Now, if we were to look back at the history of Earth, we think that for nearly half of its life, it existed in an anoxic state, meaning there was no atmospheric oxygen. And even before that, when the Earth was first formed, it was completely sterile, devoid of any life for around 750 million years. Then, about 3.8 billion years ago, microbial life first emerged, and it has stuck around since. In fact, around 80% of Earth's entire biological history has been solely microbial. And so it's only in the last 600 million years or so that we've seen an explosion of diversity on Earth. Now, some of the first cells to appear on Earth were bacteria and archaea, and crucially, they were capable of anaerobic metabolism, meaning they didn't require oxygen to survive or grow, which made perfect sense in an oxygen-free world. Then, a little while later, and by little we mean a couple of 100 million years, the first phototrophic bacteria appeared. These microorganisms had developed a way to harness sunlight to produce energy. However, unlike modern plants, they performed anoxygenic photosynthesis, meaning they didn't produce any oxygen as a byproduct. Then, nearly a billion years or so later, came a real game changer, cyanobacteria. These were the first organisms to perform oxygenic photosynthesis, splitting water molecules and releasing oxygen into the environment. And so this was the start of a massive shift in Earth's atmosphere. Over time, the oxygen levels began to build up in a slow but profound process known as the Great Oxidation Event. It was this transformation that paved the way for more complex forms of life. And so following this oxygenation, around 2 billion years ago, eukaryotic cells emerged. These cells were more complex than their prokaryotic ancestors, featuring internal structures like a nucleus and critically the mitochondria, making them much more efficient. This was a key point in history where life really began to diversify. However, for a long time life remained microbial. But then, about 600 million years ago, oxygen levels rose again, setting the stage for what we call the Cambrian explosion. This was a period of relatively short but dramatic evolutionary innovation, and suddenly many of the major animal groups we recognise today started to appear. From this, we see the rise of complex body parts, shells, skeletons, and for the first time, true predators. Ecosystems began to resemble the kinds of food webs we recognise today. From this point on, life continued to branch out and diversify. Plants colonised the land, followed by insects, and eventually vertebrates made the move from the sea to shore. Then came the rise and fall of the dinosaurs. And after their extinction, mammals began to diversify and flourish, and among those mammals, primates evolved, and eventually humans emerged. But in the grand timeline of life on Earth, humans occupy just the tiniest sliver of time, not even a pixel on the screen. And yet our existence is rooted in billions of years of evolutionary change, all beginning with those early microbial pioneers that first transformed our planet's atmosphere. Now, as we've seen, microorganisms have been here since the beginning of life on Earth, and they've colonised all areas of Earth. And so when we use the term microorganism, we're referring to living organisms that are typically too small to be seen with the naked eye, and therefore require a microscope to observe. Most microorganisms are unicellular, such as bacteria, archaea, and many protozoa. However, some are multicellular or form colonial structures, like certain fungi and some algae. But even these remain microscopic in size or have microscopic life stages. And as cellular organisms, microorganisms follow the principles of cell theory, which states that all living things are composed of cells and that all cells arise from pre-existing cells, a concept summed up by the phrase omnis cellula e cellula. This means that microorganisms reproduce by producing new cells from existing ones. And so, as an example, bacteria typically divide by binary fission, whilst yeast, a type of unicellular fungus, often reproduces through a process called budding. Now, because microorganisms are by definition microscopic, we rely on microscopes to observe them. But even under the microscope, it quickly becomes clear that not all microorganisms are the same size. In fact, their sizes vary enormously, spanning several orders of magnitude. And so to help make sense of this, we often use a logarithmic scale like the one we see here, where each step represents a tenfold increase or decrease in size. This allows us to compare everything from molecules through to acellular viruses, prokaryotes, and the largest eukaryotes in a meaningful way. So let's begin by considering mammals and mammalian cells. At one end of the scale, a human like myself might stand around 1.8 meters tall. In contrast, mammalian cells are microscopic. For example, a human sperm cell is one of the smallest, measuring around 5 micrometers in length. On the other hand, the ovum or egg cell is one of the largest mammalian cells and can measure up to 100 micrometers in diameter, just large enough to be seen with the naked eye under ideal conditions. Next, if we move on to fungi, which are also eukaryotic cells, these too cover a wide range of sizes. For example, Candida albicans is about the average for a single-celled fungus, but there are other extremes. We have Termitomyces titanicus, a mushroom-forming species with a cap that can reach about 1 meter in diameter. Some fungi can grow even larger than this. Their underground hyphal network can extend around 1 to 5 kilometers, making them among the largest living organisms on Earth, although they rarely get the credit for this feat. Then, if we turn our attention to prokaryotic cells, specifically bacteria, we again see considerable variation. Thiomargarita namibiensis is one of the largest known bacteria visible to the naked eye, while Mycoplasma genitalium sits at the other extreme, among the smallest both in physical size and genome size. Escherichia coli falls somewhere in the middle, typically around 1 to 2 micrometers in length, and it is often used as a model of an average bacterial cell. Now below this level we move into acellular entities. Viruses, for example, also vary widely. Rhinoviruses, which cause the common cold, are among the smallest. Now below viruses we reach the molecular scale. Structural proteins like collagen can form long fibrils measuring up to about 300 nanometers in length, while DNA within our own cells, if stretched end to end, would measure nearly 2 meters in length, all compacted down into a microscopic nucleus. And so now that we've explored how microorganisms vary in size, it's just as important to recognize the scale of their influence. Microorganisms are everywhere, and they've played a crucial role in shaping both our environment and our health. From the moment we're born to our end of our lives and beyond, microorganisms continue to influence the world around us in powerful and sometimes invisible ways. And on that note, let's look at some of the ways in which microorganisms impact the world around us. So first, let's take agriculture as an example. Many important crop plants, such as soybeans, peas, and lentils, collectively known as legumes, rely on bacteria to carry out processes called nitrogen fixation. These bacteria convert atmospheric nitrogen, which plants can't use directly, and convert it into ammonia, a form they can take up and use for growth. And so over time, the relationship has evolved into a close symbiosis that benefits both the plant and the microorganism. In addition to this, microorganisms also play a crucial role in animal farming, though in a slightly different way. You see, cows, for instance, can't digest much of the tough plant material that they eat on their own. Instead, they rely on specialized bacteria living in a chamber of their digestive system called the rumen. These microorganisms break down complex plant material, releasing nutrients that the cow can then absorb and use. And of course, it's not all good news. Just like humans, agricultural crops and livestock are also vulnerable to a wide range of diseases. These infections can dramatically reduce yields, threaten food security, and sometimes require drastic measures such as mass culling to prevent outbreaks from spreading further. And a good example of this is foot and mouth disease, a highly contagious viral infection that affects cattle, pigs, sheep and goats. Another is influenza, which can spread rapidly amongst farm birds and pigs, leading to severe losses and raising concerns about strains that might even jump into humans and cause disease. Next, microorganisms can also be harnessed to produce biofuels. So for example, methanogenic archaea can generate methane through anaerobic metabolism, whilst other microorganisms can ferment glucose to produce ethyl alcohol, both of which can be used as a sustainable energy source. Now, in reality, this is not practically revolutionary. What we're doing is simply tapping into microbial metabolism that already exists, or in some cases, tweaking it through genetic engineering to serve our own needs. And so it's a clever way of taking a process that microorganisms have been carrying out for millions of years and redirecting them to our own benefit. And this is a huge area of industrial microbiology, where different microorganisms play a central role across many sectors. Two of the biggest are the food industry, which we'll come on to in a minute, and the pharmaceutical industry. In industrial microbiology, microorganisms are typically cultivated in highly controlled environments called bioreactors. These can range in size from just a few litres to massive vessels holding hundreds of thousands of litres. Inside, bacteria are provided with carefully managed nutrients and conditions that simulate specific metabolic pathways. This in turn drives the production of commercially valuable products such as antibiotics, enzymes, and chemicals. But it doesn't stop there. In many cases, we don't just rely on microorganisms as they naturally are, we genetically modify them. And this has opened the door to producing extremely valuable products, one of the most famous examples being human insulin, which is manufactured using genetically engineered bacteria, replacing the outdated method of extracting insulin directly from cattle and pigs. This has essentially revolutionized the treatment of diabetes and brought the cost of treatment right down for most of the world where healthcare isn't privatized. Now, the final key industry we'll look at is the food industry, which relies heavily on microorganisms to produce a wide range of products. Classic examples include cheese, yogurt, bread, beer, and fermented foods like kimchi. However, even some less obvious favourites such as chocolate heavily depend on microbial activity during their production. But it's not all positive. Microorganisms are also responsible for food spoilage and food-borne illness, both of which pose serious economic and public health challenges. This is why food safety and microbial controls are such a critical aspect of the industry, ensuring we can enjoy the benefits of microbes in food production whilst minimizing the risk that they can pose. Next, we have the role of microorganisms in Earth's geochemical cycles. And so these cycles involve the formation, breakdown, and recycling of natural resources such as minerals, metals, and fossil fuels. And microorganisms are central players in all of them. A good example is bioleaching or biomining, where microorganisms are used in an industrial scale to extract valuable metals like copper from ores or even mining waste materials. Microorganisms are also key to bioremediation of polluted sites, and so this is where certain species of microorganisms can break down pollutants such as fossil fuels, converting harmful hydrocarbons into less toxic products. Now, this ability has been proven invaluable in environmental cleanup efforts such as the response to large-scale oil spills. Now, microorganisms aren't just useful for clearing up land-based pollution, they also play a vital role in wastewater treatment. In these systems, a cocktail of microorganisms helps break down organic pollutants, converting things like toxic ammonia into nitrogen gas, which is far less problematic and is safely removed from the process. They can also be used to degrade the sludge at the bottom of water treatment plants, producing gases such as methane, which can be captured and utilized as a renewable energy source, turning a waste problem into a valuable resource. So hopefully up to this point you can see that I wasn't lying. Microorganisms contribute to almost all aspects of life. They drive decomposition in the natural world, help with nutrient cycling and energy flow, contributing to a sort of circle of life. And ultimately, they are crucial for maintaining ecological balance, and this makes sense. They have been around for billions of years, And we've already heard about how they've helped fix atmospheric nitrogen for plants to use, but they also help with the recycling of many nutrients within an ecosystem. Now, one interesting aspect of microbiology is that because microorganisms are the earliest, most diverse, and most adaptable forms on life, they serve as our main model of thinking about whether and how life might emerge elsewhere on other planets. And so we now come on to the most influential ways microorganisms affect us, human health. Throughout history, microorganisms have been responsible for a huge range of diseases, some relatively mild, others devastating and often fatal, especially before the development of modern medicine. And so over the last 120 years or so, microbiologists have worked tirelessly to understand and control infectious disease, and we've made enormous progress. But today, new challenges are emerging. Antibiotic-resistant bacteria threaten to undermine much of that progress, and viral outbreaks like the COVID-19 pandemic have shown us just how unprepared we can be when faced with an entirely new threat. Still, it's important to keep things into perspective. While infectious diseases remain a major global challenge, the vast majority of microorganisms are not harmful. In fact, most are beneficial, playing essential roles in keeping us healthy, supporting ecosystems, and maintaining the planet's life support systems. And with that, we come to the end of this video. I hope you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.